Wishing all viewers and listeners a very pleasant day. The topic of the discussion today is aging, concepts and models. Aging procedure begins after the organism has attained sexual maturity and it ends lastly in the death of the organism. Most animals undergo the process of aging, which is also known as senescence. The branch of study dealing with old age and aging is called gerontology. Aging varies from a few days to a few years or more. Man has an average lifespan of 100 years that is set by a group of death genes. Once the juvenile period, which is a period of active growth under hormonal influence, is completed and the organism enters the adult phase, growth is replaced by a metabolic equilibrium between anabolism and catabolism. Some animals tend to escape aging by evolving a rejuvenation process. Bryozoans are excellent examples which get rid of aging effects by discarding brown bodies and thus are able to rejuvenate a new life. No amount of improvement in sanitation, richness in diet or improvement of medical care facilities can increase the longevity of mammals. Such improvements may simply increase the percentage of survivors at lower ages in the population. The process of aging is the progressive decline in all vital activities of the organism that terminate in death goes on at different rates in animals of various species and in different individuals of the same species. At cellular level, the following processes are involved. Number 1. Possible reduction in functional efficiency of non-dividing highly specialized cells such as nerve cells, muscle cells, etc. Number 2. Progressive stiffening with age of the structural protein collagen, which constitutes more than a third of all body proteins and serves as general binding substance of skin, muscular and vascular systems. It is also the substance of cartilage and tendons. It fills up the spaces between muscle fibers and between the cells of many organs, serving as the stabilizing fiber of connective tissue. In skin, a thick mating of collagen fibers gives the skin its toughness and plasticity. During aging process, collagen accumulates changes in the molecular structure that affects its integrity and function. The aging of collagen may provide an objective index for determination of biological age as distinguished from calendar age of man and other animals. Once the fibers of collagen are laid down in the body, they cannot be renewed. Collagen molecule consists of 3 strands wound around to form helices. During collagen aging, crosslinks increase in number between the different strands and different molecules. Number 3. Limitation of cell division. Hayflick in 1968 showed that as population of human fibroblasts approaches the end of its lifetime, aberrations often crop up in the chromosomes. Chromosome aberrations and cell division peculiarities related to age have also been observed in leukocytes and in liver. In man, several organs lose weight after middle age, which can be attributed to cell loss. The human brain weighs considerably less in old age than it does in middle age. Kidney also shows reduction in nephrons accompanying cell loss, and the number of taste buds per papilla of tongue drops from 245 in young adults to 88 in aged. Animal age may also result from deterioration of the genetic program that orchestrates the development of cells. As time goes on, the following changes occur. The DNA of dividing cells may become clouded with an accumulation of copying errors. The coding and decoding systems that govern the replication of DNA operates with high degree of accuracy, but the accuracy is not absolute. Certain enzymes involved in the transcription of information from DNA for the protein synthesis may deteriorate with age. All these events lead to loss of progression of cells as a result of aging and eventually to the death of cells. Now we would learn cellular changes during aging. Aging causes changes in cells at numerous levels, which can be: number 1, morphological changes; number 2, physiological changes; and number 3, subcellular changes. Starting with morphological changes. The most obvious morphological changes occurring during aging in cells may be: number 1, decline in cell volume. The cell undergoing aging exhibit reduced cell volume. Reduced cell volume and cell death results in decreased weight of organs, thereby resulting in progressive decline of body weight. Number 2, accumulation of exhaustion pigment. The exhaustion pigment, lipofuscin, yellow pigment or brown degenerations are peroxidation byproducts of unsaturated membrane lipids, that is fatty acids, and denatured proteins. These are present in nerve and cardiac muscle cells and also in other cells of the body to a lesser degree. Number 3, nuclear pycnosis. With advancing age, the nucleus becomes shrunken and stains deeply. Such a nucleus is called pycnotic, and the degenerative process is known as nuclear pycnosis. Number 4, formation of lipid vacuoles. Aging results in accumulation of small lipid vacuoles in the cytoplasm. Physiological changes. Various physiological and biochemical changes occurring in the aging cells are: number 1, collagen and aging. Collagen, an essential basic protein present in the connective tissue, becomes cross-linked and stiff with aging. Number 2, calcium and aging. In the aging cells, calcium ions are found to accumulate in the peripheral cytoplasm due to changes in the permeability of cellular membranes. Number 3, DNA and cellular aging. Loss of nuclear material and DNA damage progresses with age. Due to this, RNA synthesis and protein production also decline, leading to cell loss with age. RNA and cellular aging. There are some evidences of quantitative age changes in the metabolism of individual RNA species. Decline in transcription may result from alterations in chromosome organizations, specifically at the level of chromosomal proteins that complex with DNA to form chromatin. Protein synthesis and cellular aging. The synthesis of protein universally declines with age in all eukaryotic organisms. Aging also causes decline in rRNA and alterations in tRNA that is required for amino acid binding. Elongation factors necessary for stabilization and continued elongation of the polypeptide chain decrease with age. Post-translational modifications also occur during aging. Other biochemical changes associated with aging are increase in cholesterol and triglyceride level, increase in blood globulin levels, decrease in alkaline and acid phosphatases, decrease in cellular respiration and increase in serum creatinine. Subcellular changes. Various subcellular changes associated with aging are number 1, plasma membrane and aging. The functional capacity of plasma membrane gets disturbed during aging. Its permeability changes. Calcium becomes accumulated in the membrane. Total lipids of membranes decrease with age. The cholesterol is to phospholipid ratio in cellular membranes increase with age. Number 2, endoplasmic reticulum and aging. The amount of granular endoplasmic reticulum decrease in the cytoplasm of old cells. In the nerves of older animals and human beings, there is decrease of nichal granules, that is rRNA. Number 3, mitochondria and aging. Due to aging, the rate of carbohydrate metabolism is decreased. Aging also affects glycolysis. The mitochondria are involved in the aging process of the animals, and in old tissues, the mitochondria become degenerated. Number 4, nucleus and aging. Nucleus remains unaltered in aging cells. The change in nucleocytoplasmic ratio acts as an important index for natural senescence and death. Incidence of loss of one X chromosome in women and Y chromosome in man have been reported in aging cells. We will further discuss aging in plants and animals. From evolution point of view, it is known that those species having separate germ and somatic cell lines only exhibit senescence. Species producing by asexual means such as fission do not exhibit senescence due to natural selection operation. It has been suggested that some genes selected by natural selection for positive effect in early life may have negative effect in later years after reproductive activity has declined due to accumulation of deleterious traits that result in senescence. Lifespan of various plants and animals have been known. The table displayed on the screen depicts the lifespan of various mammals. Aging in plants. Aging and senescence in plants occur at various levels such as at cellular level, organelle level, developmental level and at organism levels. Aging in animals. Animals from protozoans to vertebrates exhibit aging and senescence. Indefinite lifespan is found in those animals which exhibit asexual reproduction and continued growth or replacement of all body parts throughout life. This is related to genetic constitution of individuals that plays a role in aging. In mammals, a relationship also exists between the ratio of brain weight to body weight and lifespan, and the proportion of life spanned in the pre-reproductive stage and reproductive phase. The more delayed the onset of reproductive life, the longer the lifespan and the later the onset of senescence. The table shown here on the screen is prediction of lifespan of modern primates on basis of body and brain weight. Moving ahead, we will now discuss aging and organ systems. With aging, changes occur in various organ systems of the body. The development of senescence is slow and progressive. It progresses at different rates in different systems and in different individuals, leading to a loss of reserve and increasing probability of organ system failure and death. Age-related changes in organ system are as follows. Number 1. Nervous system. The changes includes atrophy of the brain and increase in size of brain ventricles, loss of brain weight, that is 1400 grams at the age of 20 to 1334 grams at age of 60. Loss of white matter, shrinkage of dendrites, loss of nerve cells, accumulation of H pigment, that is lipofuscin, increase in cerebral amyloid, decrease in synaptic density in cerebellum, loss of cerebellar Purkinje cells, decline in cerebellar integrative functions, decline in learning, decline in short-term memory and recall from long-term storage. Number 2. Sense organs. The changes include decrease in the number of taste buds, decline in olfactory sensitivity, reduction in sensory cells in nasal passages, occurrence of drooping eyelids, decline in lacrimal secretion, development of astigmatism, cataract and presbyopia, decrease in aperture of iris, retinal detachment, diminished fidelity of color vision, physiological degeneration of auditory system resulting in bilateral loss of hearing, degeneration of vestibular system and increase in touch threshold. Number 3. Immune system. The changes include impairment of both cellular and humoral immunity, involution or regression of thymus gland, decline in T cell function, increase in susceptibility to infections, increase in autoimmune diseases such as inflammation of thyroid, nerve endings and testes. Number 4. Endocrine system. Decline in TSH, decline in synthesis of sex hormones from gonads and decrease in renin and aldosterone levels are the various changes. Number 5. Gastrointestinal system. The changes include atrophy of oral mucosa and degeneration of oral connective tissue, hardening of tooth enamel, calcification of tooth pulp leading to tooth loosening, reduction in salivary secretion, reduction in peristaltic activity, atrophy of gastric mucosal lining and diminished gastric digestion, impairment of intestinal, digestive and absorptive processes, loss of liver weight and decrease in number of hepatocytes per unit volume of liver, decreased motor functions in colon leading to frequent constipation and increase in colorectal cancer. Number 6. Blood vascular system. The changes occurring include diminution of heart muscle mass and increased fibrous tissue, increase in atrial volume, accumulation of lipofuscin pigment in heart muscles, degenerative calcification of aortic valves, increase in arterial intimal layer due to accumulation of smooth muscle mass, development of atherosclerosis and pulse, and increase in systole, diastole and pulse pressure. Number 7. Muscles and bones. Atrophy of skeletal muscles, decrease in size and number of muscle fibers, decline in muscle tension, increase in osteoblast activity resulting in increased bone resorption, development of osteoporosis and loss of joint flexibility due to cross-linking of collagen molecules are the various changes. Number 8. Respiratory system. The changes occurring in respiratory system are increase in volume of ducts, decreasing gaseous exchange, increase in thickness of bronchial mucus gland layer, decrease in vital capacity of lungs, increase in dead space, diminution of diffusion capacity in lungs and respiration become progressively more difficult. Number 9. Excretory system. The changes include decrease in weight and volume of kidney, loss of nephrons, decline in filtration rate, impairment of elimination of nitrogenous wastes, reduction in creatinine clearance, frequent and incontinence urination. Number 10. Reproductive system and sexual activity. The changes are menopause in female, atrophy of ovaries, enlargement of prostate glands in males, atrophy of testes and decrease in penile erection. We would learn theories on aging. Aging is not just a natural loss of life but exact process of development, though vicious. A number of factors are accountable for the aging procedure and these jointly act through a composite network of interacting positive and negative feedback system. The major theories proposed to explain aging can be generally classified into two groups. The first deals with aging at the level of the cell and is known as the cellular theory, and the second takes into account the whole body for understanding the aging process, the systemic theory or extracellular theory of aging. Various theories explaining aging are: number 1. Neuroendocrine theory. All the body activities are dependent on neurological and endocrine systems. Throughout the life, the loss of neurons and endocrine cells takes place. As a result, there occurs reduction in brain weight due to cell loss and loss of fluids and ground substance. Changes in endocrine functions result from cell loss and changes in receptors for hormones. Number 2. Immune theory. The immune theory of aging is based on two aspects, that is, age-dependent decline in the capacity of the immune system, mainly thymus-derived immunity, that is T cell or cellular immunity, and autoimmunity, when immune system starts treating autologous tissues as foreign. These conditions lead to vulnerability to diseases and abnormalities consequential from autoimmune response. Definite disease of older age are characterized by some degree of immune system dysfunction. These diseases comprises cancer, that is failure of immune surveillance, mature onset diabetes, that is autoantibody action against insulin receptors, insulin and islets of pancreas. Some vascular diseases and autoimmune effects bring about changes in proteins such as collagen and introduces errors in transcriptions, translations or post-translation fluctuations. Number 3. Somatic mutation theory. Mutations are the changes in the DNA of a cell. Somatic cell of the body of organisms develop spontaneous mutations. These mutations get included by the errors in the replication of DNA during cell division. The impaired DNA may render the cell defective in the production of essential enzymes, resulting in the cell aging or even cell death. Number 4. Error and fidelity theory. The aging results from a decline in the fidelity of all biological components. Here, fidelity refers to true reproduction of proper proteins through transcriptions and translation. The decrease in fidelity of transcription or translation is called as error and is distinct from DNA-based mutations. Error in transcription or translation results in the addition of improper amino acids into the sequence comprising a particular protein. Number 5. Glycation theory. The theory states that glucose acts as a mediator of aging. The process involves non-enzymatic reactions that take place between reducing sugars and tissue proteins and nucleic acids. The end products of such reactions in case of proteins leads to dehydrated and rearranged unalterable structures referred to as advanced glycation and products. Similar end products are produced between DNA and reducing sugars. Glycated proteins show loss of enzyme activity, inappropriated cross-linking and decreased degradation of irregular proteins. Glycation of DNA, most usually uracil residues resulting by deamination of thymine, if not repaired by excision repair and uracil DNA glycosylase, can induce mutation. Hemoglobin, collagen and Lens proteins are those proteins that undergo glycation with age. Glycation of collagen may direct to elasticity changes in blood vessels, tendons and much of the connective tissue strain of body tissues. Number 6. Program restriction theory. It is now well recognized that the genetic information depends on base sequences of DNA. Only small amount of the information in DNA is utilized by cells of various tissues. Only the sequences suitable for a particular tissue are transcribed and translated. The remaining DNA is turned off. The theory of program restriction states that aging may be due to increased turnoff of DNA, leading to impaired capacity of the cell to transcribe essential RNAs including mRNAs, though DNA segments may not be actually lost, chromatin structure seems to change with age in a way that favors restriction of DNA transcription. This might lead to a functional loss of portions of the gene library. Thus, hereby we summarize the talk. Aging occurs in all animals and plants. It is evidenced by a decline in vital activities of life. The symptoms of aging are clearly demarcated at cellular, organ and system levels, including morphological, physiological and nuclear events. There is a clear relationship between average lifespan and aging of an individual life cycle. Numerous genetic and non-genetic theories are proposed to describe aging, which have also been discussed in the